especially in the frame of a visit of Dr. Bradford, that will give us the last speech of the day. Today we will start with a talk from Ester. Ester, she studied agronomy here at the University of Costa Rica. Later she did her master's. Today we will start with a talk also in the program here from agronomy. And she will be speaking of what is the role of the seed testing lab in CIGRAS. And Esther, she is now coordinating this lab now in CIGRAS. So, Esther.
So, thank you very much. And I'm very happy to have the opportunity to share with you all our work at the lab with this presentation titled The Official Seed Testing Laboratory at CIGRAS, Providing Seed Technical Expertise and PR. So first of all, I would like to introduce you our group, which is composed by some permanent staff and also by students. Veronica and Guillermo are our technicians, and in this semester we have Gabriel, Gerald, Angelica, Andres working with us, and I am the newest member of a group. I began to work about 3 weeks ago, and so I am very thankful to all of them and also to Andres, who has just left for his PhD studies, for receiving me in such a great way. Well, our work at CIGRAS, Seed Testing Laboratory, is focused on 3 main areas, which are services, research, and teaching. And even though teaching is a very important part of our work, today I'm going to speak about services and research. Our national legislation established that the National Seed Office is the institution in charge of supervise the production and commercialization of good quality seed, and that the CIGRAS Seed Laboratory is Costa Rica's official seed quality testing laboratory. And this means that CIGRAS functions as the technical counterpart of the National Seed Office, and it provides the seed quality criteria necessary to approve or not the commercialization of the seed. And also it is stated in the law that we must follow the ISTA, or the International Seed Testing Association, or the Association of Official Seed Analysis guidelines in all of our tests. In terms of services, we have specifically in paid services, we offer physical and physiological seed quality tests. We have accredited and non-accredited tests. We have 2 accredited tests, which are physical purity and germination, both in rice, and they are accredited by the ISO 17025 standard. To quality assurance, each 4 years we participate on the ISTA proficiency test rounds, and we participate for the accredited tests we have that are germination in rice, purity, seed purity, and also the other seed determination. It's very important to us to highlight that from 2015 to 2018 we improved our performance, obtaining a rating of A in the 3 tests. And as I told you before, we also offer non-accredited tests. And here it is very important to mention that even though they are not accredited, we follow the same standards as if they were. And so for these non-accredited tests, we offer germination, physical purity, tetrasoline test for viability, and also humidity by the oven method in a variety of different crops. And also we have specific tests, for example, in coffee and in potato. In coffee we have this test that is determining the number of seeds damaged by the coffee berry borer, and also in potato we have physical analysis and also phytopathological tests. Here we have some numbers for last year period from May 2017 to April 2018. We have a total of 1,635 samples from a variety of different species, mostly from vegetables and rice, but we also have forage grasses, other grains like corn or beans, and also forestry species, coffee and potato. Also, we made a total of 2,690 tests, from which almost 1,000 were for germination of different species, and followed by the rice tests, which are rice
from Ester, she studied agronomy here.
Other seeds and red rice and rice purity. Beyond being Costa Rica's official testing laboratories, we also do research. We have some basic and applied research projects. For example, we are now working on validating this method for shortening the coffee seed germination test. We are now working on validating this method for shortening the coffee seed germination test. We are now working on validating this method for shortening the coffee seed germination test. We are now working on validating this method for shortening the coffee seed germination test. We are now at other stage that we do, because we do it until a cotyledon expansion, so it lasts near 60 days. Now we are working on the data analysis and also compare them with the official results, so to finish the pre-validation process. Many of an important part of the research that has been done on our laboratory has been focused on the digital image analysis for automated assessment of germination by using this germination software package. And the use of this technology allows us to study germination in a more detailed way. For example, it allows us to quantify parameters such as maximum percentage of germination, time to achieve 50% germination, and some uniformity parameters. We have worked with rice, carrot, and also with pitaya or dragon fruit. Sorry. Recently, some of our co-workers at CIGRAS published this paper on seed storage, germination conditions, and germination assessment approach for seed germination of pitaya. Do you hear me? For seed germination in pitaya, and they found that using conventional and image analysis approaches to a high coefficient of germination when measuring seed germination. So this means that using this image analysis method is a efficient and a feasible method for studying the germination process in pitaya. And so, besides using this germination software, as I mentioned before, they found that it could allow to calculate additional parameters like the maximum germination, and also this is the time to achieve the 50% germination. And for example, in this graph, we can see that a water potential of -0.5 megapascals negatively affects its vigor, but did not affect the maximum germination. And also here, we can see that the water potential inducing molecules, for example, PEG or sodium chloride, also affect seed vigor. For instance, using PEG increased the T50 compared when it used sodium chloride. And also this year we began a project in Macau palm that is Acrocomia aculeata, or as it is known in Spanish, coyol, together with this university experimental station that Fabio about it. And the aim of this project is to study different factors that affect Macau palm seed germination and initial seedling growth in Costa Rican accessions with agricultural potential. This focus on two main objectives, which are characterization of morphology and color of fruits and seeds, and also by studying the effect of different chemical and physical treatments on germination. Also, we want to determine if there is a relation between this morphology and color of the fruits with the germination process. But this is just starting this year. And so finally, for our future research perspective, we want to work together with the Costa Rica Coffee Institution in determining the optimal coffee seed storage conditions for Costa Rica commercial coffee varieties. And also we would like to work with hormone profiling of Acrocomia as the germination process under different chemical and physical treatments. And also another thing we would like to work with biomarkers for seed germination quality in rice and also in common beans. And so thank you very much for your attention. If you have any questions. Thank you very much.
Well, thank you, Ester, for your very nice talk. Now, I would like to introduce Walter Quirós. Walter is the director of the National Seed Office, and today he will be speaking of the role of this institution. Something important is that Walter, he studied agronomy here also, the University of Costa Rica. He did his graduate thesis in CIGRAS, actually, and he has accumulated over 39 years of experience in the National Seed Office. And now, as director, he has been 13 years. So now, thank you again, Walter. We are looking forward to your talk. Nada más un detalle. Debido a la participación del doctor Bradford, estamos en la actividad en inglés, pero igual, si ahora Walter va a hacer entonces una mezcla entre la presentación en inglés con la va a explicar en español. Pero igual, al momento de preguntas, si quieren hacerlo en español, también no hay ningún problema. Muchas gracias.
Bien, muy buenas tardes. Un gusto compartir con ustedes, doctor. Bueno, yo quisiera iniciar haciendo una referencia al entorno que tenemos dentro del sector y los retos que tenemos en el sector agrícola nacional e internacional, y cuál es la participación también de las semillas dentro de esos desafíos que tiene la agricultura. Esto para luego resaltar la importancia de la calidad de semillas ante esos retos. Bueno, es de todos conocidos que tenemos desafíos importantes dentro del sector agrícola, como el de la globalización y con ella la competitividad que implica, competitividad que conlleva la necesidad de mejorar la eficiencia productiva, y dentro de esto el factor calidad es determinante. El tema de seguridad alimentaria, en una población mundial que crece aceleradamente y que obviamente demanda mayor producción de alimentos, y el tema de cambio climático, que nos dice que esos incrementos en la producción de alimentos deben hacerse, pero bajo condiciones adversas, y lo cual implica una adaptabilidad de las variedades que se desarrollen a futuro. También quiero poner en contexto que el tema de semillas es un tema cada vez más complejo. Es un tema que contempla toda una serie de aspectos que incluyen temas de legislación, propiedad intelectual, temas fitosanitarios, temas de biodiversidad y acceso a la biodiversidad, temas de biotecnología, políticas, el tema de la industria de semillas, de recursos fitogenéticos, de convenios comerciales, temas de calidad, de certificación de semillas, etc. Es un tema tremendamente complejo y que no puede visualizarse, o es difícil visualizarlo desde un único aspecto, un único elemento. Dentro de todos esos aspectos, de toda esa diversidad de temas, la legislación es algo que tiene que ver directamente con el tema de calidad de semillas. Y es que la expansión de la agricultura, la evolución del mejoramiento genético, la emergencia de la industria de semillas, el desarrollo de una comercialización de semillas, una comercialización formal de semillas, conllevar la necesidad de establecer reglas en el comercio, reglas en la producción, el uso y el comercio de semillas. Específicamente, la legislación, en el contexto de la legislación en el cual nos desenvolvemos en el país, contempla no solo la legislación de semillas y la reglamentación técnica conexa, sino también temas de legislación fitosanitaria que tienen que ver con el movimiento de germoplasma, el movimiento de semillas, que tienen que atender requerimientos de tipo fitosanitario en esta legislación. Temas de semillas también están considerados dentro de la legislación en materia de agricultura orgánica, sobre todo el tema de semillas criollas. El tema de biodiversidad tiene que ver con aspectos de acceso a germoplasma, tiene que ver con temas de propiedad intelectual, incluyendo también convenios internacionales como el Convenio de Diversidad Biológico. El tema de biotecnología también, a través del protocolo de la biotecnología moderna, el protocolo de Cartagena sobre la biotecnología moderna, que es la que regula este tema biotecnológico en el país. El tema de recursos fitogenéticos, que tiene que ver con el acceso, el movimiento de germoplasma y lo que se llama dentro de este contexto el acceso facilitado a recursos fitogenéticos para alimentación y la agricultura. Y el tema UPOV, el tema de derechos de propiedad intelectual para variedades vegetales. Existe en Costa Rica, bueno, Costa Rica está adherido al convenio UPOV y también tiene una legislación de corte nacional, que también, bueno, en este caso la Oficina Nacional de Semillas aplica la legislación en materia de derechos de propiedad intelectual, así como la legislación en semillas propiamente. Esta legislación tiene que ver con la regulación, es el marco legal para la regulación de la actividad de semillas en materia de producción, comercio y uso de semillas, bajo una perspectiva o un concepto de protección al consumidor, o sea, protección al agricultor, y para propiciar una sana competencia en el comercio de semillas. En términos generales, si yo quisiera resumir en una frase cuál es el objetivo de la ley de semillas, es el de promover la disponibilidad y uso de semillas de alta calidad. En eso se resumiría una legislación de semillas. Esta legislación tiene como funciones generales la promoción y organización de la producción y uso de semillas de calidad, el promover o propiciar un adecuado abastecimiento de semillas, el establecimiento de un sistema de control de calidad de semillas destinadas al comercio Y señalo destinadas al comercio porque se refiere al comercio formal, no al comercio informal que existe a nivel de ferias de semillas u otras de tipo de semillas criollas u otras. Y establecer las reglas y los mecanismos de control para la circulación y comercio de semillas. Esto se hace a través de 4 mandatos o 4 instrumentos que tiene la ley de semillas. Uno de ellos es el control de calidad de semillas, principalmente a través de la certificación de semillas, el registro de variedades comerciales, que vamos a ver rápidamente ahora de qué se trata, las regulaciones para la producción y comercio de semillas y la protección de derechos de propiedad intelectual para nuevas variedades. Este último es a través de una legislación adicional a la ley de semillas, que es la ley para la protección de obtenciones vegetales. Aquí cabe preguntarse tal vez que por qué el Estado, a través en este caso de la Oficina Nacional de Semillas, establece una regulación en el comercio de semillas. ¿Cómo se justifica esta regulación? Bueno, hay varias razones. Primero, que el agricultor es un consumidor y como tal tiene derechos a una semilla de calidad. Cuando una semilla es comercializada, se genera también una responsabilidad de parte del comercializador. Por otro lado, todos sabemos que la calidad de la semilla no puede ser detectada a simple vista. Eso podría ser engañoso. A veces hay semillas que tienen un aspecto excelente, pero no sabemos cómo está la germinación. A veces sabemos la germinación, pero pueden haber problemas de vigor o pueden haber problemas de otro tipo. También sabemos que la semilla es uno de los pocos insumos vivos que se utilizan en la agricultura y por lo tanto están sujetos al deterioro. Aunque la semilla es un vehículo o un mecanismo de transferencia de tecnología, esa tecnología que se genera, se desarrolla a través del mejoramiento genético, y es un paquete genético que se ha desarrollado a través de programas de investigación, la semilla también es uno de los principales vehículos de plagas. Por otro lado, tanto el que compra como el que vende la semilla deben conocer características de este elemento. Por ejemplo, el valor agronómico, que incluye una serie de aspectos que vamos a ver, y la calidad de la semilla. También, bueno, las reglas del comercio favorecen una sana competencia, como ya mencionamos, y la semilla es definitivamente un factor determinante del comportamiento, del resultado de un cultivo. También es bueno saber el ámbito de aplicación de esta legislación, quiénes son los actores que participan dentro de un sistema de semillas, que son los que están, a los cuales se les aplica todo lo que es el sistema de control de calidad. Bueno, dentro del sector semillero tenemos al sector privado, en donde participan las compañías de producción de semillas, que tenemos las tradicionales, las industrias tradicionales de semillas, por ejemplo las arroceras, productoras de industria de producción de semillas de arroz. Tenemos las compañías biotecnológicas, tenemos las productoras de almácigos. Por otro lado, tenemos también dentro del sector privado a importadores y exportadores de semillas. Vamos a ver algo de eso en cuanto a la magnitud o la participación de estos actores dentro del abastecimiento de semillas. Distribuidores locales, que son los agroservicios que encontramos a nivel regional, y otros actores como entidades público-privadas, llámese Corbana, el café, Laica, dependiendo del cultivo. Tenemos en algunos casos programas de certificación de semillas, como el caso del café. Tenemos también el sector público, el sector estatal. Aquí están dos entes regulatorios como la Oficina Nacional de Semillas, como el Servicio Fitosanitario del Estado, un ente de innovación y transferencia de tecnología agropecuaria, y tenemos una industria nacional estatal, el Consejo Nacional de Producción, que participa básicamente en la actividad de semilla de frijol y maíz. Está la academia, las universidades, que son entes de investigación, de transferencia de tecnología, y también en alguna medida de producción también de semillas. Y está el CATI como un ente, como un centro internacional de investigación y enseñanza, y el usuario final de todo este sector, que son los agricultores. ¿Cómo se abastece el país de semillas? Bueno, hay abastecimiento a través de producción nacional y también de importación. Siempre señalamos el caso de los vegetales, en donde más del 90% de la semilla que se utiliza en el país es importada. Ahí es loable la labor que realizan, sobre todo la academia, que viene realizando algunos trabajos de investigación y desarrollo de nuevas variedades. Lo hemos tenido en el caso de Chile, en el caso del tomate, que son iniciativas, aunque aisladas, pero nos parecen una muy buena iniciativa que nos permite demostrar que sí se puede producir semillas de vegetales en el país, y podría esto llegar a tener un éxito tan grande como lo ha tenido el caso de la papaya, que es otro de los ejemplos. También importamos semilla de maíz, un alto porcentaje del área nacional, que por cierto es muy poca hoy día en el caso del maíz, pero un alto porcentaje de esa área es de semilla híbrida importada. También forrajes, se importa desde hace muchos años una cantidad muy significativa de semillas forrajeras, principalmente de Brasil. Y luego tenemos la producción de semilla nacional. Nosotros somos autosuficientes en semilla de arroz, frijol, papa, café, caña de azúcar, una serie de frutales, raíces y tubérculos. Sin embargo, hay que señalar que no necesariamente hay programas de certificación formales en todos estos cultivos. En algunos de ellos sí, como el arroz, el frijol, el café y otros, pero no necesariamente en todos hay programas formales de producción y comercio de semillas. Y hay que señalar que en todos los cultivos somos dependientes de la importación de germoplasma para mejora genética, en todos. Tenemos programas de certificación en arroz, frijol, maíz, café, palma aceitera, cacao, aguacate. Y tenemos programas de verificación de estándares. No todo es certificación, obviamente en los cultivos, en las semillas importadas, lo que se hace es una verificación de calidad de los estándares de calidad que están establecidos. Básicamente es en maíz, en hortalizas y en forrajes, es donde más se realiza ese tipo de muestreo para análisis de calidad. Esto lo quiero mencionar, son dos factores diferentes, lo que es el registro comercial y el concepto de calidad. Sin embargo, creo que es necesario mencionar, porque es un tema muy importante, el tema del registro de variedades comerciales que maneja la Oficina de Semillas, en donde toda persona física o jurídica que quiera comercializar semillas de aquellos cultivos en los que existe un régimen de registro comercial, tienen que inscribir las variedades ante la Oficina Nacional de Semillas. Esa inscripción necesita que el interesado tenga que demostrar lo que llamamos el valor agronómico. Valor agronómico lo componen una serie de aspectos como la productividad, adaptabilidad, resistencia a enfermedades, calidad industrial, el ciclo del cultivo, etc. Y el otro aspecto es la descripción varietal, que básicamente es valga la redundancia, una descripción de las características fenotípicas de la variedad que nos permitan identificarla con claridad. Eso tiene que demostrarlo a la persona que quiera poner en el mercado una nueva variedad. Y esto es un ejemplo el registro de variedades comerciales. Esta cantidad ya fue superada, son más de 500 variedades de diferentes cultivos que están inscritas en el registro de variedades comerciales y que son de diferentes cultivos. La mayoría de ellas son hortalizas, pero también en cultivos como arroz, frijol, maíz, forrajes, café café, etcétera, están inscritas en este registro comercial. La labor propiamente de control de calidad que se lleva a cabo en el país se hace principalmente a través de la certificación, que es un sistema integral de control de calidad que se desarrolla en las diferentes etapas de producción de una semilla y que permiten verificar el cumplimiento de estándares de calidad que están definidos en una reglamentación técnica que contempla una serie de aspectos relacionados con la fuente de semilla, las categorías, los requisitos del terreno, con requisitos a nivel de campo y de laboratorio. Esta verificación que se hace en esta trazabilidad de la producción se hace a nivel de campo, inspecciones de campo, y luego se hace a nivel de laboratorio, que ya aquí se contemplan estos diferentes factores de calidad que se valoran a nivel de laboratorio. Perdón. Este sistema de verificación de la calidad o de control de calidad es un control que hace el Estado, en este caso a través de la Oficina Nacional de Semillas, y vamos a ver ahora también la participación del CIGRAS dentro de esto, en la parte del laboratorio. Pero el Estado es un fiscalizador de la calidad de las semillas. La responsabilidad de la calidad de las semillas no es de la Oficina de Semillas, no es del CIGRAS, es del comercializador. La calidad la tiene que reunir o la tiene que cubrir el comercializador de semillas, y el Estado lo que tiene es una responsabilidad de fiscalización, de verificación, que está cumpliendo con estándares de calidad establecidos oficialmente. En el caso de la semilla importada, como mencionaba anteriormente, lo que se hace es un muestreo de la semilla que se importa y un control de calidad a nivel de laboratorio específicamente. ¿Cómo logramos saber cuándo entra semilla, cuándo se importa semilla? Precisamente porque la ley de semillas establece un requisito para la importación, que es el registro de la importación o exportación de semillas. Eso es por ley y no puede obviarse, digamos, ese requisito. Toda persona o empresa que quiera, o institución que quiera traer semillas al país, tiene que cumplir con un registro de la transacción que va a hacer. Ese registro está alineado con el Servicio Fitosanitario del Estado y con aduanas, con el Ministerio de Hacienda, de modo que una semilla que llega a aduanas no puede llegar sin un registro de importación o con un certificado fitosanitario. Y eso está alineado, no se autoriza la importación o la exportación si no están cumplidos los requisitos tanto de la Oficina de Semillas como del Servicio Fitosanitario. Llegado a este punto, quisiera destacar algo que tal vez pareciera más político que otra cosa, pero en realidad es algo que venimos trabajando y que vamos a mencionar ahora en la siguiente lámina, un trabajo coordinado con el CIGRAS, con el Laboratorio de Control de Calidad, y que culminó el año antepasado con la Política Nacional de Semillas. Es una visibilización de la actividad de semillas en el país, un trabajo de poner en relieve la importancia y el valor estratégico de las semillas dentro del sector agropecuario. Esto ha venido escalando, ha venido adquiriendo cada vez más importancia, al punto que el tema de semillas es considerado, es incluido hoy día dentro del Plan Nacional de Desarrollo. El Plan Nacional de Desarrollo es toda una estrategia nacional de desarrollo del país en las diferentes áreas, y dentro de estas, dentro del sector agropecuario. Dentro del sector agropecuario consideran algunos temas relevantes para ser promovido por el gobierno, y uno de esos temas es el Programa Nacional de Control de Calidad de Semillas. que ya se viene incluyendo dentro de ese Plan Nacional de Desarrollo. Y se establece como objetivo controlar el cumplimiento de las normas oficiales de calidad de semillas demandadas en el mercado para una mayor competitividad del sector agropecuario. Se establecen también metas anuales de control de calidad de semillas. Esa meta para el año 19 es de 4.677 toneladas métricas de semillas que se someten a control de calidad. O sea, ya es una cifra importante de semillas. Bueno, ahí va incluido todo, semillas como arroz, como el caso de granos, que incluyen un porcentaje muy alto de este tonelaje, pero también hay que considerar que dentro de este tonelaje están todo el control de calidad que se realiza a las semillas importadas, o sea, toda la semilla hortícola, la semilla de maíz híbrido, la semilla de forrajes, que también están incluidos dentro de esta meta. Entonces, bueno, este es un factor importante que ha permitido resaltar la relevancia de la calidad de semillas dentro del sector agropecuario. Yo quiero señalar, ya para ir enfilándonos al final, una alianza estratégica que se establece entre la Oficina Nacional de Semillas y el Centro para Investigaciones en Granos y Semillas. Esta es una alianza que ha estado durante los últimos 40 años, una alianza que se establece dentro de una legislación, y es que la ley de semillas, la ley actual, es una ley del año 1978, establece que los análisis de calidad de semillas, los análisis oficiales de calidad de semillas, serán llevados a cabo por el laboratorio oficial que es el Centro para Investigaciones de Granos y Semillas de la Universidad de Costa Rica, el CIGRAS. Esta es una alianza en donde la Oficina Nacional de Semillas hace las tomas de muestras de toda la semilla que se somete a control de calidad y se remite al laboratorio del CIGRAS, absolutamente toda. Una semilla solo puede tener un análisis oficial si es enviada por la Oficina de Semillas al laboratorio del CIGRAS. Y así se ha hecho durante 40 años. Ya vimos ahora en la charla anterior el volumen, la cantidad de muestras que realiza el laboratorio año tras año, que anda en el orden de 1.500 o 2.000 muestras, no sé, por ahí. Por lo menos son más de 1.500 muestras al año. Entonces, esta alianza ha sido determinante, ha sido una simbiosis que ha funcionado perfectamente durante estos años y que, bueno, no podemos dejar de mencionar esa importancia de esta alianza. Finalmente, igual, parte de esta alianza nos permitió trabajar durante 2 o 3 años en el desarrollo de una política nacional de semillas, que fue una iniciativa conjunta del CIGRAS y la Oficina Nacional de Semillas, y que luego se incorporaron otras instituciones y que culminó con la oficialización de una política nacional, fue a inicios del año 17, sí, y que nos permite tener esa política, esa guía de lo que debe ser o de la orientación que debe llevar la actividad de semillas en el país, y que contempla una serie de elementos que están señalados aquí, el tema de generación de variedades como fundamento de cualquier programa de semillas, el tema de producción de semillas propiamente, la extensión agrícola, mercadeo, desarrollo de empresas semilleras, la seguridad en materia de semillas, la generación y fortalecimiento de capacidades y la plataforma legal. En todos estos temas es la visión que se tiene a futuro para lo que es la actividad de semillas en el país. Y existen pocos países que cuentan hoy día con una política nacional de semillas. Finalmente, agradecerles con una frase que me encontré hace unos años en el CIGRAS y que me ha aprovechado de esa frase de Alvin Toffler y con la cual creo que todos estamos de acuerdo. Muchas gracias. No sé si alcancé a preguntar. Muchas gracias.
Now we will have a presentation from Víctor Jiménez. Víctor, he studied agronomy and he did also see his master's here in the University of Costa Rica. Then now we will Víctor, he studied agronomy and he did also see his master's here in the University of Costa Rica. Then now
Thank you, Luis. Thank you, Dr. Bradford, for coming to Costa Rica and to the University of Costa Rica. It's a pleasure to have you here. And now I'm going to present the work, part of the work we do at CIGRAS, specifically at the Plant Biotechnology Lab. And my presentation aims to specifically at the Plant Biotechnology Lab. And my presentation aims to specifically at the Plant Biotechnology Lab. And my presentation aims to specifically at the Plant Biotechnology Lab. And my presentation aims. We are facing a problem, and this is there's going to be more people to be fed in the future. But if you take a look at some numbers, we can see that there's enough number of calories being produced right now to feed the actual population and even more than the actual population. But there are some limitations that have to be taken into account, and this is like the use of crops that should be used for food, for other purposes like biofuels. There are losses in the field and post-harvest. There are problems, serious problems in the distribution. We have a high percentage of food being wasted because of plant consumption, and additional reasons like disasters, conflicts, and so on. But even with all these limitations, we can achieve this requirement of 1,800 calories per person per day in the future with actual cropping area and also techniques that are being used right now. But if you go into more detail, not only thinking on number of calories and amount of calories, we should focus on malnutrition that's unbalanced and inadequate intake of nutrients, both macro and micronutrients. And we have the following consequences like undernutrition, overnutrition, and what's so-called hidden hunger. And the former two you cannot stem very easily because of overweight or obesity or other symptoms. But hidden hunger is more difficult to be noticed, and this is a situation that affects a large number of people, 2 billion people in the world, and that's not, as I already mentioned, it's not very easily noticed, and something can happen, that's so-called double burden. That means that there's people that they are not, they can be even obese or overweight, and they are suffering from deficiency, especially on micronutrients, on essential vitamins. How can the hidden hunger be fought? One option is food fortification during processing. That's being done in Costa Rica and Central America with vitamin A and other micronutrients and vitamins. In the plant breeding process, you can focus on biofortification by selecting and making process with the aim of enriching our crops or our grains with minerals and vitamins. You can add supplements, which is necessary in several cases, like in pregnant women and so on. And you can increase diet diversification, exploring new food sources. And this is what we have been trying to do with some of our projects. If we see on some FAO numbers, we can see that there are 250,000 plants that can have an agricultural potential, but less than 7,500 from them are being currently used. And if we go deeper, our world food production depends on 150 plant species, but 12 of them provide 3/4 of the world food, and only 3 so-called mega crops provide 50% of the calories in the world. With these numbers, you can imagine which could be the situation in case of viral disease for one of these crops, or facing climate change as well. So, to increase food security in a sustainable way in the long term, it's necessary to think on including additional plant species in the human diet. And the tropics are especially prone to as a source of these plant species because of the large diversity that can be found in our regions. And following up with this idea, I would like to present you 3 of our projects from the projects we have been running the last years in our lab. I would like to focus on the domestication of purple pitaya, Hypocereus is the genus, on our work with beach palm, batteries, and aiming at reducing the vitamin A deficiency. That's not an issue probably in most Central American countries because of the enrichment of our food, but in other regions in the world, and also in the clonal propagation and selection of papaya by means of, in our case, by means of somatic embryogenesis. In the first project, which is the one with pitaya, with the dragon fruit that was mentioned earlier, this is a climbing cactus. It's native from our region, and the main issue of this cactus is the fruits. Because they are very attractive, they have been produced in several countries, not only in the region, but also in Asia, Israel, Vietnam, Thailand, and we have different species, and we have different varieties or genotypes. And in terms of increasing food diversity and also thinking on functional foods, the phthalates are maybe a key issue here, because there are pigments that can be used to substitute artificial colorants. We started working with pitaya about 17, 18 years ago, and we tried to approach comprehensively. We first started with the characterization of the fruits, of the genotypes, and also selected tissues of the plants. Then we proceed with some thinking on potential uses, evaluating market potential, industrial applications. After that, we focus on selection and propagation, and parallelly we conducted some other studies. And in this first step that we started many years ago, we focus on the characterization in cooperation with School of Food Technology and with the colleagues in Germany of different genotypes, and also on the ripening process and the pigments and the antioxidant profile and the phenolic compounds, and that could have a potential because of the functional properties. We also found that calcium oxalate crystals were present not only in that time. So we decided to analyze also these crystals because they are an issue. If you want to promote consumption of a new crop, then you should be sure that it's safe to consume it. So we found out that the crystals are not in the pulp of the fruits, then we can recommend the consumption of the pulp. We also identified different shapes, crystal shapes, and we found for the first time, I think, that the presence of both monohydrate and dihydrate crystals in single tissue. Okay, in the second steps, the potential uses of pitaya and pitaya products was also evaluated, and in this sense it's important to focus a little bit on the particular pigments that are found in pitaya fruits, which are betalains, as I already mentioned, which have a similar hue than anthocyanins, but they are more stable on high pH values, and that they can be used for coloring of milk products and similar products. And this could be an opportunity to substitute artificial colorants in such products. The common betalain sources are mainly from red beet, but there are some drawbacks from red beet that are present in pitaya fruits. So we think that pitayas have a potential for the food industry. And some works were developed in this sense. First with extraction of the pigments, or the extraction of the pulp, separation of the seeds, and also the analysis of the cell wall and the composition of the mucilage. Also to look for the utilization of byproducts from a potential industry, pitaya industry. Also the seeds, another byproduct, could be used as a source of oil. And after having identified the properties of the fruits and also a potential for their use, industrial or as industrialized or as fresh fruits, the logic is if you want to improve the use of this crop, you need to have enough planting materials, and that was not the case with pitaya because it's a crop that has not developed in the country for many years. Traditionally, pitaya is very well known, but was not part of our diet for many years. So we work on the propagation of pitaya. Usually pitayas are clonally propagated from adult plants, and this is because the seeds, the plants developed from seeds have a longitudinal period, and also because of cross-pollination, you never know what's going to come out from crosses. And clonal propagation is usually done through cuttings from field plants, but these cuttings are very bulky and is not very efficient to propagate this way. So we developed a protocol for the in vitro propagation of pitaya from pots, and this is more friendly to the plants because less damage is caused to them. The propagules are less bulky, and the propagation rate is also much higher. And this was already mentioned by Ester, and the reason for starting seeds in spite of the drawbacks I already mentioned is because of the breeding programs and also for germplasm conservation. Okay, parallel studies focus on different aspects. One was the induction of callus cultures that was not reported before, and these callus cultures could be used as a model to study the synthesis of the betalains, because in the fruits you have to wait for normal development of the pigments, but if you But if you can find elicitors that can be used to precisely induce the synthesis of pigments, and you can follow up the process in terms of biochemical process, but also in gene expression and so on. We initially managed to induce the synthesis of the pigments, and we have to work further on this topic. Since in our lab we have a long lasting experience with chloroplast isolation, culture, regeneration in different plants, we also started working with the isolation of chloroplast from Hyoscyrus and from Semillas to genera of vine cacti, and the aim will be to increase the diversity in terms of pigments or colors available, and also in terms of potential for the food industry, because related genera have different flavors besides the already mentioned different colors. Okay, these are some of our results from the chloroplast tests. We have been able to isolate chloroplast from some tissues. We also did some work in the framework of thesis on the main diseases of Italy and Costa Rica, since very few commercial plantations were available in the country. In the natural environment, diseases are usually not an issue, but as soon as you start with the monoculture, then you see some problems. Trying to advance on the taxonomy of the plants, we also did some work. We are working with molecular markers, because much of the characterization has been done with physical descriptors, and they are very viral, and you can find very high variability and changes according to the climatic conditions. The flowering process has been also an issue for us, and we started doing some characterization, anatomical characterization of the bud development, and at the same time with the analysis of some genes that could be participating in flowering determination. And the practical issue with this is if you think on Italy as a commercial crop, we have a window of flowering, a window of production, and we would like to expand this window and to have more productive months along the year for the growers to take advantage of higher prices. And with a model of genes being expressed before you can see any development on the tissues, then you can advance on factors that could be defining the gene flow reduction. We have also some other ideas in mind regarding the research on Italia. And the second project has to do with the peach palm. Peach palm is a neotropical palm that is found naturally from Central America down to central Brazil, and it's being used mainly for fruits, which is restricted to some countries in Latin America. Not every country in the region is used to the consumption of the fruits, and also because of the heart of palm, which is a very expensive product in the world market. Focusing on the fruits, the peach palm fruits have very high contents of carotenoids, but not only that. Compared to other sources of carotenoids, the peach palm has the advantage of also having very high lipid contents. And because carotenoids are lipid soluble, then this increases the bioavailability of the carotenoids, and then the intake into the bloodstream is much higher than in other fruits. And even with, you cannot read this very well, but even with, we found out some numbers with among the diversity of peach palm genotypes can be found in the country. From the very large and orange fruits, you can supply the recommended daily intake of vitamin A by taking 3 to 5 fruits. And since the fruits are very starchy, you can process them, you can extract the flour out of the fruits, and you can, without having to create, have it change in the diet of the people, you can include a source of vitamin A into the diet. And mentioning the diversity, the large diversity that is present in not only in the country but in Latin America, it's important to work with the genetic resources. And the two largest germplasm banks in the world are located in Costa Rica, but they are very old plants, and they started to die, these plants, so it's important to clonally propagate these plants to preserve the diversity already present there, because the plants are cross-pollinated. And the approach we started was also the tissue culture by means of somatic embryogenesis through thin cell layer culture, and we developed a protocol that we are validating with different accessions, different genotypes. And the novelty of this protocol is that for the first time it was possible to propagate by this means adult plants, because all efforts before were conducted with seedlings, and with seedlings you already lost the genetic background. In the case of papaya, also this is not underutilized, unknown crop. Some of the properties of the papaya have been investigated before, and there is an increasing consumption and production of papaya in the world. This is also a crop that's originating in the region, or the center of diversification is in Central Mesoamerica and the Caribbean, and it's very relevant in terms of micronutrients and like vitamin C and provitamin carotenoids. And papaya, the huge diversity that's present in the country and the region, allow to identify some genotypes that are interesting from the health point of view. And in one of these works, the focus on the carotenoids, it was observed that the lycopene is more efficiently absorbed from papaya than from tomato. And tomato is the typical source of lycopene that is what's mentioned in the literature, and also it's broadly mentioned. And likewise, in the case of beta-carotene, the vitamin A carotenoid, this is more efficiently absorbed from papaya than from carrots. And in additional study, we focus on the post-harvest and ripening of the crop, and we induced or modified the ripening process by treatments with an inhibitor of ethylene reception, and then with ethylene by itself, and we took a look at the gene expression of different candidates, genes that were related to the fairness of the fruits, and focusing on the clonal propagation of the papaya plants, trying to avoid the fact that more than 3 or maybe 4 plants have to be planted, and the seedlings should be planted in one spot for hermaphrodite plants to develop there. Then through somatic embryogenesis, you can clonally propagate only the desired genotypes. Even if this is an F1, from an F1 population, you always have this sex segregation in the F1. So this is a possibility for clonally propagate desired genotypes and be sure about the sex of the plants. Okay, this is the group at CIGRAS. It's a picture from last year, and thank you for your attention.
Well, thank you, Víctor. Well, now I would like to introduce to you. After that, he took his master's degree in the same university, but in this case it was horticulture. Then later moved to the University of California, Davis, where he got his PhD in plant physiology. And then from then he started having a very fruitful career, moving to Australia, then going back to US, go back to Davis, getting a professor position. Then later, very important, he founded the Biotechnology Center and also Centro. That will be the topics that he will be speaking. Just since 2013, he got the recognition of distinguished professor at the University of California, Davis. Thank you once more.
Well, first of all, let me thank you for the invitation. I appreciate being here. I wish I could address you in Spanish. You'll have to suffer with my Texas-based English. Hopefully you can understand me. But it's a delight to be here. I'm going to give you mainly a report on our Seed Biotechnology Center and a program called Seed Central that we created. Having come here and heard already today a great many things, I'm struck by the similarities between what we're trying to do with different crops and what you're trying to do here. So hopefully what I have to say will be helpful for your process here in strategic planning and thinking about the future, which I think actually we can do together. So a little about our Seed Biotechnology Center. Our theme, I guess, is at the top. The science, agronomy, and economics of plant biotechnology converge at the seed. We said this quite a long time ago, but when you think of the commercialization of biotechnology, nearly all of it has been through seed companies to date. I think that will be changing as we see strategies for using cells and other strategies for using biotechnology. But so far, it's been seed companies that have had the opportunities. Our Seed Center has a long mission, but I'll read it. Our mission is to mobilize the research, educational, and outreach resources of UC Davis in partnership with the seed and biotechnology industries to facilitate discovery and commercialization of new seed technologies for agricultural and consumer benefit. It's very long, but you see our focus is on innovation and research, but also on commercialization and delivery of those products to society. So I'll cover both of those aspects. So you don't get lost, I'm going to give a quick overview of our sort of founding and the highlights of what we've done in the last 20 years. I'll then come back and address some of those topics more explicitly, current programs that we have there, and then we'll have a discussion of what should we do next. Our programs are having a transition to the future as well, so we can discuss that. So we started the Seed Biotechnology Center in 1999. We started a small program. I was a seed biologist. I'm basically the only seed biologist at UC Davis. And we have quite a robust seed industry in California. And yet we do not have very many specific programs, and any specific programs addressing the seed industry. So we thought we should start a program that would interact with that group. So we started that program in the College of Agricultural and Environmental Sciences. We created an advisory council. We offered our first course. And importantly, we started getting early funding from a industry-based group, California Seed Advisory Board. It's a board established by the state of California such that all seeds sold in California pay a small tax. They pay an assessment into a fund to support the state seed lab and to support research and those types of activities. We were the first program where they would start funding research, so we had some convincing to do with the seed industry that they should spend more money on research, but they did that. It's helped us a lot. Along the way, we were able to raise some other funds, such as for a plant transformation facility. We did not have one at Davis. This is early days, still 2002, 2001. But part of our fundraising campaign, we raised money to develop a place to be. We also got the plant transformation facility at Davis opened up. As we started to grow, we got a director of research. We started hosting workshops. And we did raise some money that allowed us to join into a building that was being created and put our funding into that. So we moved into that building in 2003, started doing research there. A lot of our research was, and you'll see, is on genomics, DNA-based markers, developing marker-based breeding technologies, particularly in vegetable crops. Our focus here is more on fruits, ours are more on vegetable crops. Developing marker-based breeding technologies, particularly in vegetable crops. Our focus here is more on fruits, ours are more on vegetable crops. Developing marker-based breeding technologies, particularly in vegetable crops. Our focus. Some other research, gene flow, for example. When certain genetically engineered crops appeared, there were concerns about movement of pollen and seed quality, seed integrity, and so on. We did work on that. Published bulletins on that in the early days of Roundup Ready alfalfa and so on. In 2006, we created the Plant Breeding Academy. That's a major program of ours, and I'll publish bulletins on that in the early days of Roundup Ready alfalfa and so on. In 2006, provide advanced training Plant Breeding Academy for breeders. It's a major program of ours, and I'll publish bulletins on that in the early days of Roundup Ready alfalfa and so on. In 2006, advanced Plant Breeding Academy for it's a major program of ours, and I'll publish bulletins on that in the early days of Roundup Ready alfalfa and so on. In 2006, were very valuable at the time. Before sequencing became so cheap, microarrays were very useful for assessing variation, for marker testing, and so on. We've hosted various international symposia along the way. We'll talk a little bit more about that as well. Just promoting seed research in general. International symposia. So by 2008 or so, we were doing work on seed traits. So my own work is mainly on the seed quality traits. We've worked on molecular biology of those. We started developing tilling resources, for example. That is mutation-based approaches, because the regulatory environment for commercialization of biotechnology in the vegetables is still very difficult. So we went back to working on more mutation-based resources. We also, on the seed side, started working on some seed respiration research, which I'll describe a little bit later. My colleagues did more work on we also, on the seed side, we also have done specific studies on plant breeding curriculum needs, things that the seed industry is suggesting that breeders need to know now. So by 2010, we had enough of an education program that we needed a director of education. So we have a director of education programs now. We expanded our plant breeding academy. It started only in Davis. Now we have a European branch. I'll say in a moment, we have an African branch as well. We've initiated work about that time on seed try. I'll mention that. And we've started offering other business courses in seed quality, seed business, that I'll discuss a little bit later as well. So, 2011, you can see that this is sort of a history of technology and biotechnology. We're moving along with different technologies. More recently, done work on haploid induction. One of my colleagues at Davis, Simon Chan, developed a very interesting system to potentially make haploid inducers in almost any crop. It's in H3 gene, so we've done some work on that. More gene chips and so on to assess genetic diversity. 2011 and 12, we created Seed Central. This is an externally oriented organization that I'll describe a little bit later in partnership with us. We have this external organization called Seed Central to create closer ties with the seed industry. Work continued on resistance genes in pepper, spinach breeding, and so on, more on seed respiration. This is a program I'll describe a little later, which is a corporate affiliate partnership program. It's a program we created specifically to encourage companies to support research on our campus. So we're trying to lower the cost, the overhead cost that they pay to sponsor research with us. I'll describe that a little more later. 2013, we started our African Plant Breeding Academy in collaboration with the African Orphan Crops Consortium. So I'll come back and describe that. Pepper and spinach genomes were sequenced in our laboratory using new techniques, particularly in the latter. One of the first uses of certain sequencing techniques was in spinach. So more recently, we've been working on the dry chain concept, which I'll describe. Very useful here. I hope we have a chance to discuss this strategy with you. More workshops on seed analytics, and we also started a program called Collaboration for Plant Pathogen Strain Identification. So started in collaboration with the seed industry to develop collections of tester lines of a crop and the corresponding pathogen strains, so that when people advertise they have resistance to strain 3 of some particular disease, everyone agrees what they're talking about. So it's for standardization of the naming of resistance genes. So it's a separate program for that. 2015, an outgrowth we did not have, really. We had a large breeding effort at Davis, but not a plant breeding center. So the plant breeding center started new technologies, for example, bulk segregate analysis, fairly old technology, but with direct sequencing. So we demonstrated its effectiveness in lettuce. Color genes identified in pepper, so that is very greener peppers, greener spinach, those types of things. 2016, all along, I have been working on mathematical modeling of seed germination. I didn't describe that here, but that was a project we've had all along, and we applied that to ecology as well. We offered a course on seed production. And we've identified that certain seed dormancy-related genes also can affect flowering, which I think is very interesting for those of us involved in both the flowering and the seed production. So this highlights that we host various meetings. We hosted the International Society for Seed Science Conference, National Association of Plant Breeders Conference, and so on. And particularly in pepper, again, my colleague is finding loci of interest for various important traits. Last year, a group of people there, Davis and others, demonstrated nitrogen fixation by maize. That's pretty important, I think. We'll see if it can work, but it is actually happening in Mexico in the field as we speak right now. So it's a remarkable finding, I think. Hosted more conferences, and Seed Central received a community partner award that I'll mention a little bit later. So this year we're going to celebrate our 20th anniversary, and a highlight is that I get to come to CIGRAS and visit with you here as well. So just a quick overview. That's sort of the history of our creation, but also I think the history of the technologies, the development of these different biotechnological approaches, and also a lot of plant breeding. So let me go back and go into a little more detail on each of these topics of what we've done, what we're doing now. We've had research projects in all of these crops and areas, from seed technology through to breeding and genomics of these various crops. Current projects along these lines, pepper breeding. My colleague Alan Van Dynes, the Director of Research, is doing pepper breeding. Working on various topics. Mechanical harvesting is important for us. Labor cost and availability are becoming limiting for us in California in some of these crops. So mechanical harvesting is something we really need. Fruit shade, horticulture, virus resistance, and so on. He's also working on spinach breeding. We have a particular issue with downy mildew of spinach in California right now. We are every time we put a new resistance gene out, it's overcome almost immediately by the fungus, which is breeding itself. So it's a very difficult situation. I mentioned the maize microbiome and this nitrogen fixation. I'll come back to that in detail. Carrots, we don't do carrot breeding, but we collaborate with Phil Simon, a breeder in Wisconsin, who's the primary carrot breeder in the U.S., and do a lot of genomics and marker work with him. We have a current project on melon quality, somewhat similar to what you were mentioning before of texture, flavor, phenotyping, transcriptomes, metabolomics, and so on, to try to identify melons that are longer lasting on the shelf, crispy, but highly flavorful. That's the goal. My colleague Alan Van Dyne's worked a lot with cotton from very early days, so has been involved in all the cotton genome sequencing efforts, gene editing, haploids, and so on. My own work is more on the area of the seed germination area. We've worked on a lot of work in lettuce on high temperature dormancy, light responses, G by E effects, genetic type approaches to identifying loci involved in these traits. More recently, we've been working quite a lot on multispectral and digital imaging for seeds. How can we use that? You saw there already, you're using some here. It's a very interesting area for potentially identifying seed quality determinations, also upgrading, that is, putting it on sorters and so on, upgrade seed lots. And also been working on drying technologies. I'll mention that as well. So we have a lot of projects currently going on. This is one I mentioned about maize. Very interesting project. Was funded primarily by Howard Shapiro, who's the Chief Agricultural Officer of Mars Incorporated, if you like M&Ms, that's from Mars. Howard was living in Mexico 40 years ago, in the Oaxaca region, and saw this corn growing, 4 meters, 5 meters, 6 meters tall, 40 fertilizer, 3 years ago, stuck in his mind. And he came back and he's funded a lot of this research. So there's something unique about the genotype and also the roots. These genotypes of corn have a lot of adventitious roots at the nodes. They secrete this gel, which cultures a microbiome and also sufficiently lowers the oxygen levels so that they can fix nitrogen. So this paper was actually demonstrating that they do, doing nitrogen N15 tests, other types of rigorous tests to show that this corn is fixing nitrogen in symbiosis with this corn. So the question, of course, is can that be reproduced in corn that's not 4 meters tall? It's more like commercial corn, but it's very early. I will say that this was very widely commented on, was the top tweet for the whole University of California last August. That's a new criterion, I guess. Did you get the top tweet? My colleague Alan works on pepper breeding, and this is just an example of mechanical harvesting. So there have been harvesters developed for peppers, but not very good. And largely because we've not really worked on the plants that we need to be harvested. California produces about 30% of all the processing tomatoes in the world, all mechanically harvested, because 50 years ago we developed both the plants and the machines that made that possible. We had to rebreed the plant and we had to rethink the machinery. So that's what we're doing right now with peppers, is to rethink the plants, and particularly Alan has found traits where the peppers, particularly these type, jalapenos and so on, will come clean at the stem. So the stem will easily break off at the fruit when you're harvesting it. And so this is the difference between currently available varieties mechanically harvested and these over here. So real potential, I think, for at least those fruits. Moving that to bell-type peppers may be more challenging, but at least for some of these more industrial-type peppers, it looks possible. I should say, if you have questions now, I'm happy to stop if you want to follow up on these. Some of the more recent things we've been doing on seed germination, seed biology. A few years ago, we published on this gene. It's a very interesting gene called delay of germination. Martin Corney's group started working on this identified this as a genetic locus important for dormancy. We've looked at it in lettuce, particularly. We found that it is involved in dormancy of lettuce, high temperature dormancy of lettuce. And in the process of those studies, for example, we silenced that gene, and we found that it caused very early flowering. So this is the control plants of lettuce, very quick flowering, the same gene. So here we're, to me at least, it's quite interesting that we have the same gene that is working in the dormancy of the seed pathways, also working later in flowering. This gene was thought to be only expressed in seeds, so how could it also be working subsequently in flowering? But in fact it does. And this paper actually suggests that one of the ways it does might be through microRNA pathways. That is, they're very powerful microRNAs that control seed dormancy, control flowering, and regulate a whole series of transcription factors and genes downstream from that. My own goal with this is that could we develop this, since it's microRNA based, could we develop this into something that could actually be applied? That is, could we put on microRNAs, perhaps stabilized with nanoparticles, for example, to get them into the plant and switch flowering on command. So for example, we were working on breeding of spinach. We currently produce spinach seeds 3 places in the world: in Denmark, in New Zealand, and in northern Washington, in north of the US, because it requires long days to flower. So we're stuck with the trait of that plant inherited through history, restricting where we can actually grow seed, because we have to grow it where we want it to flower. Of course, we don't want it to flower when we're growing it for spinach. We want it to be vegetative the whole time. So we have this contrast between selecting for vegetative growth, then having to make seed, or make them flower. So I think there's a lot of opportunities, not only with this strategy, but with others, as you mentioned, looking at the genes controlling flowering, so that we could have a lot more control in the crop production, but also then, when we're ready to make seed, say, please flower now. For example, nicking in hybrids, getting the males and females to flower, and so on. It's a great opportunity, I think, for the next coming years. How can we get more control over flowering as well? And we've done some other things. I mentioned this project before of identifying mutants, unknown mutants, using bulk segregant analysis and whole genome sequencing. This was a case brought to us by a seed company. They had a mutant that had traits they wanted, not really germination on the fruit, on the flowering, that's not a good trait, but came along with it. This worked very well. We were able to use this strategy and identify the mutated gene in one stroke, just in one cycle, pretty much. I mentioned we're working on seed technology. There are various ways to do this. We've been working with this particular instrument. It's developed in Denmark, called a video meter. Inside here, it has LEDs that can take pictures in 19 individual wavelengths. So you get one wavelength per picture. You take the whole series, you get a picture of your seeds at all 19. And they also can now put in 3 different filters, so you can get fluorescence. So you have quite a combination, 19 inducible wavelengths, 3 filters for each of those to look at the fluorescence wavelengths. So it's a very powerful instrument. And by combining those different wavelengths in different ways, you can see a lot of things. This is, for example, tomato seeds. And if you're a seed tester, these little blue ones are not tomato seeds. So I mean, you can get purity analyses, or you can identify other species and so on. These were some squash seedlings. People wanted to identify cracks. If you get the wavelengths right, the cracks just jump out at you, and so on. Different things on these are bean seeds and so on. So we're looking at various ways to use this technology for analysis, and then also potentially for separation. The key would be to take your recipe you've learned here of the wavelengths and how to combine them, put that into a machine that will sort seeds and just put them into categories and upgrade seeds with that. Another strategy we've taken is on oxygen. I won't go into great detail, but this is an instrument came out of the Netherlands. It allows the respiration measurements on individual seeds. So we can put one seed in a well, put a cover on it, and it has a little dye, fluorescent dye on the top. And then it shines a light on that, gets the fluorescence back, and the fluorescence is proportional to the oxygen content. It was actually developed for beer bottles. You put a dot of the dye inside the beer bottle, it goes by, a light shines on it, and it says, is it sealed or not? Does it have low oxygen inside or not? If it's transparent, you can get an immediate read. A lot of uses for it, but they did develop this instrument here where we can put seeds on plates or tubes. The machine will go and automatically measure them as much as we want. So it's robotic, it's very efficient. I won't go into the details, we've kind of published that, but we find a very tight relationship between the respiration behavior and the germination behavior, which probably makes sense, but it's very nice to know. Seed by seed basis, we can actually substitute respiration measurements for germination measurements with pretty high quality R squares and so on. It's a very useful approach. So what we're trying to do now is combine the last two things I said. Can we image seeds initially, keep track of them, and then measure them for their germination respiration aspects so that we could sort them on a digital basis with the image rather than germination after the fact, which is destructive. So we would have a non-destructive way to pre-screen seeds, knowing that we would also be selecting for physiological traits. So that's the type of thing we're doing. Here's a lot of data, I don't need to go through it, but this is a parameter related to the speed of germination, and this one is the seed size. So it's a very simple comparison. It would make a lot of sense. If we look at that, these were seeds, tomato seeds that were screened just like a standard seed cleaning operation, different size screens. So you get seeds of different sizes, that's the different colors. And in this direction, they're spread out in terms of their germination rates. So for tomato seeds, it doesn't matter a whole lot. You don't really gain a lot with size. That is, the small ones are just about as good as the big ones, except maybe a few over here. But that's the type of thing we're looking now at. Other traits, blindness in brassicas. If you grow brassica seed, a lot of the seedlings, the meristem doesn't grow, so you have a blind seedling. How do you screen that out of seeds and so on? So we're trying a lot of different characteristics to say, can we use these new digital imaging strategies and assess seed quality with it? At the very end, I'll talk about an even more interesting, exciting approach to this. We're just working with the company with. The last one I just mentioned is the dry chain. I mentioned we've worked quite a lot on drying. This was a USAID funded project that is an international development project, really focusing on how do we dry and store seeds in humid climates, like yours, where it's warm all the time and it's humid most of the time. That is not a good situation for seed storage. When you come to grains and so on, it's also not a good situation for mycotoxins, which I know you're the experts here as well. So I'm going to try to learn from you on that. But humidity and warmth are the keys for that as well. So what we did, we worked on different drying strategies that I won't go into great detail on, but we developed what we call the dry chain, which is very simple. It just needs to harvest, dry the material. Most seeds in the world are dried in the sun. We have air drying systems as well. We were working on a desiccant-based system. That is, we use a desiccant like silica gel, but more powerful, to just dry the seeds so that we can do the drying in humid environments. You just have to get the seeds packaged away from the environment and then put the desiccant with it. So we can talk about that later. But then after that, the important part of this part of these humid parts of the world is after that they have to be packaged and stored, which of course seed people have known for a long time. Seeds being shipped into the tropics often are in metal cans or are protected in some way. Grains are not. So if those grains are sitting out in high humidity, there's a high potential for fungal growth, mycotoxin, and so on. Keep them in those containers. That's the key. There are crop storage bags used in a lot of these tropical regions, but the idea is the farmer will use those to store grain and then dump it back out when they market it. It goes back into the environment again. We say, let's keep it dry. And then it stays good. So basically, this is what we term that is the dry chain. If you work in produce, you're probably familiar with the cold chain. Anybody heard of the cold chain? If you have lettuce, you have to chill it, keep it cold all the way. Complete chain of command all the way to the market. Apparently, nobody thought that that also applies to dry products. Dry chain, it's so simple. But nobody thought of it, I don't think. But it's really what we need. It's just like if you have lettuce that's been chilled, you don't just set it out for a day or two in the air and then start up again. It's the same with seeds and things you need to keep dry. You have to keep them continuously dry, or else they will take moisture. So this is a whole separate project, recent paper we've got describing that, and some very interesting tools like pH paper for humidity. I could show you this later if you want. That is just basically paper that turns pink or blue depending on the humidity, making it very easy in the field to assess the moisture content of your product. Really very straightforward. So that was a bit on our research. I'll go into our education programs in a little bit more detail. These are some of the courses that we currently offer. These are what are coming up this year. So, for example, we have 2 offerings of our Seed Business 101. I'll tell you what that is in a minute. We're offering a seed production course. This is a very interesting course we've offered for quite a while now, 24 times. We call it Seed Business 101, which just means elementary seed business, first course in seed business. It's targeted to people in seed companies that are new to the seed industry, which is almost everyone. How many people grew up thinking they're going to work with the seed industry? Almost nobody. It's not a very visible part of the agricultural industry itself. So what we do is we have industry experts teach this. Our instructors are currently mostly retired CEOs of seed companies who want to pass on their knowledge. And it allows people who are new to the seed industry to come and get up to speed very quickly. It's a very popular course. And we offer 2 types, one for agricultural crops, one for horticultural crops, because the seed business is somewhat different in the 2. Here's a new one we're offering this year, hemp breeding and seed production. Well, last December, it became legal in California to grow hemp, which means cannabis with no THC, very low THC. So it's mainly for these other compounds, CBD compounds and so on, but it's legal. It's a total scramble. And nobody knows a lot about some of these things, the reproductive biology, genetics, and so on. We actually have seed certification system for this, but no seed growers yet. So it's very early. So we'll be offering a course like this. So we try to be available to offer courses that are supportive of the seed industry. Our Plant Breeding Academy is our sort of classic program. It's an intensive program, 6 weeks of instruction over 2 years for commercial plant breeders. So come every 4 months for a full week. So you get 6 full weeks of training. Genetics, very heavy on statistics, very heavy on changing breeding programs into being marker-based, genomics-based breeding programs. It's not biotechnology, it's not recombinant anything, but it's just marker-based breeding. So we're now in our 7th cycle of that at Davis, in our 5th cycle in the EU. We offer it in the one in Davis. All classes are in Davis. This one is in 5 different locations in Europe, the 6th one in Davis. I think that would be a great opportunity if you have some breeders here. 5 different locations. Each location we visit breeding programs in that area. It's a very good program. And the next class begins in October in Europe. These are no more than 20 people, so we like classes that are small enough that you can become very intensive between the instructors and the students. We'll come back to that in the future as well. I mentioned this a while ago. This is the African Orphan Crops Consortium. This is also supported by Howard Shapiro from Mars Corporation. Howard became aware of the nutritional problems you mentioned, hidden hunger and stunting. He became aware that 30 to 40% of children in certain areas, India, Africa, are stunted from poor nutrition. He said he decided he had to do something about it. So one thing that he did, because he's a breeder by training, he started the African Orphan Crops Consortium, which was to sequence indigenous African crops. So we're going to sequence 100 indigenous African crops. That is, these are crops that are grown there. They're had very little breeding effort. Orphan, that just means very little breeding effort. We're sequencing those. We're sequencing 100 genotypes of each of those. So it'll be about 10,000 genomes by the time it finishes. But it will completely enable marker and genomics-based breeding in these crops. Howard realized that if he did that, he would need a breeding program of African breeders who could use it. So that's where we came in. He came to us and said, what about offering a plant breeding academy? So this is different. These are not younger breeders training up in methods. These are full-fledged, PhD, nearly all PhD, public breeders, most of them in government programs, coming to learn these more modern techniques and start expanding their programs into some of these additional crops. Most of them are working on commodity crops for their main job, but they have an interest in some of these other crops. So we're now in our class 4. This is the 4th class we've done. We have 36 participants out of 600 applicants. The first time we had 30 participants out of about 60 applicants. So this has really gotten to be the thing to do in Africa. This particular class, for example, has 23 countries. It's almost equal men and women, and they all have PhDs. So this is a very high-level thing. Already, graduates of this program, the first 4 programs, have created an African Plant Breeding Association, which is having its first meeting this summer. So this is going to be transformative, I think, for indigenous crops. They have the same, this has the same goal that you mentioned. We have crops that people eat that are nutritious, that just need some basic breeding work to solve the basic problems, to make them much more available for people. So the goal of this program is to graduate 150 African breeders working on those crops. We've just added a 6th class. We thought it would go 5 classes. We'll go 6th class totally on permanent crops, trees and other permanent crops. So you may have some interest in that, what may come out of that as well. So this is just in our courses. So this is our calendar for this last year of all the courses we offer. So we're heavily invested in providing ongoing education for the seed industry primarily. So that's a major effort of what we do. We also, as I mentioned, have a plant breeding center. This really is an organizing group of the plant breeders on campus. They are focusing much more on the internal training, that is graduate programs, undergraduate programs, and so on. So now we have a concerted effort on our attracting more people into breeding programs, maintaining our education programs in that as well. So they're taking care of most of that. We're doing the external. And this is my colleague, Alan Yananich, who also is Director of Research for me and Associate Director for that. He's a busy boy. Public service, another area that we're invested in, same as you mentioned before. Research, education, public service, and I'll add outreach at the end. These are some of the public groups, institutions, government institutions, the USDA, APHIS, FDA, industry groups, American Seed Trade Association, California Seed Trade Association, Council for Agricultural Science and Technology, an NGO that publishes papers and so on. So we work with all these groups as we try to be a scientific resource for all these groups. So we try to be there to analyze the situation, give them feedback as best we can. So for example, this is a fairly recent paper that I contributed to with this CAST group, Regulatory Barriers to the Development of Innovative Agricultural Biotechnology by Small Businesses and Universities. That's us. We've done a lot of great things. People have made all kinds of interesting plans that will do a lot of great things. We can't give them out because of the regulatory situation. If you're not a big company with deep pockets, you will not get through the regulatory system in the U.S. and certainly not. If you don't get through the U.S., it's very difficult to get through it anywhere else. So we're trying to at least put that issue out there. We do things, as I mentioned, like hosting international meetings. This is one for International Society for Seed Science. Two things. One, we like to support these. The other, we feel this is a service to our members in California. If we bring major international meetings to California, many more people can attend can often travel. So this is a service, I think, to our students and also to seed companies who can send multiple people to a local meeting, but might only send one person to an international meeting somewhere else. So we try to bring meetings to California as much as we can. Last year, the Cucurbitacea breeding conference was hosted by us, for example, another example. This is this program I mentioned before, the Collaboration for Plant Pathogen Strain Identification. This was initiated by the American Seed Trade Association, mainly by the vegetable seed producing companies, to address the issue of advertising what strains your variety is resistant to. When you have multiple strains of a given disease, and if it means a different thing in one country than another, it's a big problem, because the grower will buy it thinking that they're going to get resistance, and then it doesn't work. So what this program does is funded by industry. The companies, seed companies contribute, and Phyllis Himmel, the plant pathologist, she runs the program. They collect the tester strains of the crop and the pathogen strains that when you apply them, you can see the variable response, the resistance response among those, and from that you can categorize them into the right category. So it's fairly straightforward, but this does all of that. It collects all the seed, it collects all the pathogens. You can just order from them if you want to start in a crop and they have it. You just, okay, send me the whole package and I can get underway, compare my lines against those, get into the breeding program. So it's just an example of, because we had this program, they decided to locate this in California with us, and we're happy to have them there. Communication, the last area I'll talk about is communication. We have an annual report every year. I brought a bunch of these. I meant to bring them over here today, but they're in your office. But I have about 10 of these. I'll leave them with Louise so you can spread them around. Just an example of annual reports we do. We try to be in the news as much as we can. That is, if someone, again, a reporter or someone wants an opinion on a current topic that's in our area, we want to be the ones to respond. There are often many others willing to respond that don't have the same opinion as us, so we'd like to be contacted. So we try to be available. This was in Chilean. You know better than I do. What is that? The Chilean article about this topic. Now I'm going to mention Seed Central. This is a big outreach effort for us. This was really created, I'll go ahead and put a picture up here. This was really created in collaboration with Francois Korn, who is the owner and founder of SeedQuest. SeedQuest is a website that is sort of the global website for the seed industry. So if you don't go to know SeedQuest, go to seedquest.com. What he basically did was he just did a map of seed companies in the California Seed Association in California. This is Davis. There's a cluster here. There's a big cluster down here in Semillas where the vegetables are grown. So the breeding companies cluster around the university and also the fields. It's a very productive agricultural area. And here, the same thing, very productive for the vegetables. But what he worked in the internet and Silicon Valley world, and he realized that we had an industry cluster, but we were not exploiting it. It would be as if people in Silicon Valley were working totally independently. They didn't know that there was another technology company just down the road. That's not the way they work. Those technologies build on each other very efficiently down there. So what he said was, why don't we start a networking system? We have nearly 900 firms authorized to sell seed in California. We have 80 members within an hour or so, and it's about a 3-hour drive. And we have many people who drive up to our meetings from there. And most of the major seed companies, global seed companies, are operating in California, so they have branches there. So the idea was we joined together, we create Seed Central, which is an external nonprofit organization. So we have this alliance with them. We find that there are certain things we really can't do from within an academic environment. There's certain things they can't do being an external NGO. So we've created this partnership. For example, members join Seed Central, pay a small fee to become a member. That helps to fund that side. It helps to fund some operations for us. We can't charge memberships like that in the same way, quite the same way. So it's a very symbiotic relationship. The idea is to energize the seed industry, facilitate communication. This is one of the main things we do, is just provide an opportunity for communication back and forth. We want that to lead to research collaboration between the industry and the university. And we want to make this region even more attractive. If you're a young person anywhere in the world and you want to work in the computer world, you might think of going to California, to Silicon Valley. You might eventually end up there if you join a company. We want to be the Silicon Valley of seed. If you want to go to where seed is most exciting, come here. It may not be that yet, but that's what we would like to say. If you want to be where the center of the world of seed is, we should come here. Which has its own multiplier effects. That is, once that starts to happen, you get the additional groups, you get the suppliers of seed companies, and you get other types of companies coming to build that up. Of course, it provides jobs for our graduates. They don't have to go. They can come and they can get a job when they walk out the door. So, some of the events we do, in fact, our next meeting, we don't do it during the summer, but our next meeting will in fact be our 20th anniversary celebration. I don't know who that guy is, but we can, if you can join us for that. So every month, the key to this is every month from September through May, second Thursday, we're going to have a meeting. Francois realized that's important. That way people can put it on their calendar, they'll know it's going to happen, they'll show up. Francois predicted, he says, you watch international companies, their members will start planning their travels to California around these meetings. And it happens. Because they know if they come on that day, they'll see all kinds of other people. They'll get to mix with a lot of other seed people. What we find is that industry people in our area very much appreciate the opportunity to come together in a neutral space at the university and talk to each other. They're not going to give away any secrets, but they're going to talk to each other. And Francois tells the story, for example, we had a program speaker talking about flavor. Flavor, very important in almost every kind of crop. He overheard two people talking. They said, okay, you're in strawberries and berries, Driscolls. You may know Driscolls, strawberries and berries. Another one from a breeder of onions. They said, we're never going to compete. Onions are not going to compete with your strawberry market. But we're both interested in flavor. Let's talk. So they did. So they started sharing resources in the equipment, the flavor approaches, the chemistry of flavor of compounds, and so on. So this is the type of synergy that just starts to happen when you start having regular meetings. The other things that we do, internship and student programs, very heavy emphasis on that. So students in the University of California, Davis now, who are undergraduates or graduates, if they routinely come to our meetings, they nearly always have a job when they leave. They've already met people in the industry. They already know them. They've already got a relationship with them. We have trips with them to go visit companies. We have the soft skills courses. When you go to dinner with a CEO, how do you act? Do you know how to network in a room? If you go to a room, do you know how to go up and talk to somebody? These are the soft skills, they call them, in human resources that we work on. We do research forums. We also do ag tech. These are all the exhibitors that were in our recent ag tech thing. These are new technologies and so on, startup companies from our institution and others that we're exposing the industry to. Here's what we did on, I mentioned this corporate affiliate partnership program. Basically what this is I won't go in detail, but it's a mechanism by which we can promote consortia research. Two or more companies come together, they can get a reduction in the overhead costs for doing research with us. So basically it's an incentive to come and do research with the university and to do collaborative research. So we're happy that last year this Chancellor gave us an award, Innovative Community Partner of the Year for Seed Central. So in other words, this has become a model at our institution for how we can interact with our surrounding industry. We have an enormous food industry around us that's not doing this. Seed is very small compared to the scale of the food industry. But we're trying to expand this sort of approach now out to the bigger tomato processing industry, rice industry, almond industry. We don't really work with the fruit trees because we're seed mainly, but in the bigger picture with food, we're starting to try to do that. I just mentioned that here, the World Food Center. It turns out for the last 2 years, I've been the interim director of a program called the World Food Center at UC Davis. So this is to try to pull together the resources we have, again, mobilize those resources for innovative, sustainable, and equitable food systems, bring together these partners, address this food problem that you mentioned. We have a lot of people to feed coming up very soon. It's going to be very challenging. And we do have a big resource here. So I'll just show, this is our resource just in our College of Agriculture and Environmental Science. We have about 15 departments and more than 60 food-related centers in health, policy, food systems. My seed center is one of those. But there's 60 more. There's 59 more, all the way from consumer, food technology, nutrition, breeding, animal science. There's all the way through. We have all of these areas of food that we have a resource that we need to exploit. So that's our staff that works on this. I give them all a lot of credit. So now the next thing, what's next? So I'll fairly quickly go through this and we can have a discussion because you see where we are, so I just wanted to put some topics up on where are we going next, or what could we do. I'll start from the other way back. Outreach, it looks like we need to get more involved in some of these areas. It's not going to be me, but now we have people in our staff, younger people who know how to do social media and so on. If we're not there, we're invisible. So I think, in fact, we're going to have to get more involved with that. We're on Facebook, but it takes effort. It takes, particularly in these, if you're not there frequently, you get disappeared. So you really, it's a resource you have to invest in. We are working with others. For example, there's this new website sponsored by Bio, which is the Biotechnology Industry Organization in the US, all the bio for medical and so on. But they're doing a series of videos and so on, particularly focusing on trying to get a more positive public view of gene editing. So they came and we did a video today was about lettuce, for example, to show why would we want to use gene editing? Well, there's many things that we could do with that. Why, just to try to put a scientific base, a positive face on it. Public service, big area for this. These are things that we've done. A lot of effort on regulatory input. You see a lot of papers we've tried to write on summarizing the situation in the regulatory, trying to advocate for allowing us to do certain things in crops that are not giant crops, that is in other crops. We've done a lot of that, but I think we need to do more of that. Labeling will start in the US next year. We will have to have this bioengineered label on it. We have not had that. That's going to be a big issue of how that gets rolled out, implications for us. We need to be more visible, write more opinion pieces, reviews, and so on, conferences. We're sponsoring a conference in November with our environmentally oriented center at UC Davis. We're asking the question, what do we do? We're a scientific institution. What do we do when the public will not give us the social license to do what the science tells us we have to do? So if we think of climate change, we know we need to make some changes. And yet, the public is very resistant to doing those changes. They just tried to raise the gasoline price and the petrol price just a little bit in France. Huge yellow vest protests came out, just the most minimal thing. So we have this issue that the science is very clear. The climate change science is very clear. Society is not ready to go there. We're going to use that in GMOs as another example of where the science is very clear. Every major scientific organization in the world has said that's no more dangerous than what we've already done. We've mutated plants until they die. And yet we do not have the social backing to use those technologies. So I think we have to think about it because we don't have infinite time. We don't have infinite time with both the climate change and the looming population to come at us. So these are some of the things that we think we can do. In education, a big question for us, should we go to electronic courses, these massive online courses and so on? It takes a lot of travel to come to the courses we teach. They're international classes, people travel a lot and so on. We have not really gotten into electronic distance learning yet ourselves. We like the fact that we get people together with the instructors and so on. It's clearly something we have to consider though. Moving bodies around all over the world is not the most ecological thing to do either. So we have to start thinking about some of these things. We're going to keep focusing on internships and research opportunities for students. Our main driver really in a lot of what we do in this area is the future employees for the seed industry. So that includes all the way through, from young to graduates and so on, and existing employees. The seed industry, biotechnology, is it's a skill set that you can't learn once and know forever. Workers in the seed industry are going to have to be retrained every 5 to 10 years. I've gone through at least 3 cycles of totally transformative technologies in my career, and that pace is increasing. So keeping a workforce up to speed with the technologies we know and we want to employ, and you get the top tweet. It's a considerable amount of education beyond graduate school and so on. It's an ongoing process. And then research. I thought I'd stop here where we could discuss this more as we go on, but so many things are possible here. I'll just list a few. For us, gene editing will be critical because that has been essentially deregulated for certain applications in the US, so now there's an opportunity to use that more. We'll see how it gets accepted in the market and so on. I think gene editing is limited. I think we still need transformation. I wish we had called it gene grafting. It seems less threatening to me than transformation. But we didn't do that, but that's what we're doing. We're grafting genes. But at any rate, I think we need both, but that's my view. I mentioned before, I think we can do a lot in this control of flowering, seed development, and so on. This whole area of using genes from whatever part of biology, putting them in microbes to make specific products is already booming. They do not encounter a lot of the regulatory burden we do with plants. If it's an ingredient, if it's a processing aid, there's various ways that it doesn't have to be labeled as a GMO, and you don't have the huge regulatory burden. So that part of the industry is growing very quickly. Of course, in medicine, it's totally gone that way. But in food, it looks like there's going to be a lot of opportunity there, too. I mentioned this digital analysis. Combined with artificial intelligence, this is going to be amazing. So I mentioned just the one project. I mentioned the project before on digital imaging and so on. Here's a company in Israel, SeedX. Anybody heard of SeedX? It's a new company in Israel. They're combining these. The founder of the company helped create face recognition technology. Sold it to Facebook. So when you post a picture and it names who that is, it's using that technology. If you're in China and you go to get on the airplane, you look at the boarding thing, it captures your face right then. So this is very powerful technology. But this company said, well, what if we pointed that to seeds? What if we looked at seeds? And they're claiming that, in fact, you can learn more than you might think. That is, it may be possible to score for traits that you might not expect simply by scoring them with digital imaging backed up by very complex algorithms that artificial intelligence can create. So you train them on a certain comparison you want to make, and then you can put a lot of seeds on there and it'll tell you which is which. I don't know if it'll work. I'm not here to justify it, but I'm interested enough. We're going to get one of these machines in our lab. They're going to place a machine that we're going to try some tests on and see if it will work. But I don't think we should sell artificial intelligence short. We don't really know how powerful that might be, so I'm not going to sell it short. So I'm going to stop there and say thank you very much for having me here. Hopefully have shared with you what we've been doing, and I'm going to be here the rest of the week to learn more about what you're doing and how we can cooperate and work together. Thank you very much for having me. Okay, I've taken up most of the time. If you have questions or discussion, I'm happy to.
When you partner with private companies, what type of services is it these companies look for them in the university? What questions do they want to answer? What type of research do they want to do in conjunction with SDC?
Yeah, that's a very good question. What type of research do companies want to do with us, or that we will accept to do? For the most part, we try to pick projects that are pre-competitive, meaning it's more basic than companies want to do for themselves, but not so basic that they can't see any application. A good example would be, they're not very interested in funding a lot of Arabidopsis research. Very great research, but until you demonstrate it in a crop, they're not going to put their money into it. So that type of thing, where technologies that have been demonstrated, for example, in Arabidopsis or genes or approaches that you can apply in a crop, they're very supportive of it. or that's what we find, the more progressive seed company. Can you put it into their germplasm? Or can you give them, we do a lot of genetics to identify interesting traits. They will very quickly, and sometimes they support that, they'll quickly go back and screen all of their germplasm for that to see if that's useful for them and so on. And by getting consortia together, a lot of the genome sequencing, virtually all the genome sequencing I mentioned were supported by consortia of companies. Because you could do it by yourself, and it's gotten cheap enough now, but before it was quite expensive. But there's not a huge competitive advantage if that's then made publicly available. But it enables a huge amount of marker-based research and so on to get underway. So I think that's the trick for finding the sweet spot with companies, is find something that is too big or too far out for a single company to do. That's the value of bringing in a consortium. If you get 5, 6, 7 companies together, it becomes doable. If you get 10 or 15 companies together, it's cheap. Why not? Why be left out? Join in, because it costs you almost nothing. So those are the types of strategies we use to get them to buy in. So basically, it's funding research that we want to do. It's funding students, graduate students on projects to get finished and so on. Turning out new employees for them. And finding that spot where it's not so basic that they can't see an immediate application, but it's too risky. So it's kind of a de-risking also. I mean, we're de-risking to say, yeah, now you can take it from here. And that's where we don't get any useful. In other words, we can publish that, but if it's just another application in another thing, then it gets less and less valuable for faculty. So there's a little sweet spot there. Does that make sense?
Following this question, do you regularly have feedback from the companies, like to say, like, okay, can you do this project, or can we do this, or it is more use cases that says to the companies, could you please support us on this, or how it goes, in which direction?
Well, I mentioned up here, I won't go back to it, I mentioned the Vegetable Research Forum. So 2 or 3 times, every other year, every third year, we sponsor a meeting to bring the research directors of the major vegetable, any vegetable seed company who wants to come together for a brainstorming session. We ask exactly that question. What can we do for you? You know, what's limiting you now? What do you need some research on? In consensus, try to get them to think about it. And then, of course, we turn around and say, well, let's get a project together. We can do that. I mentioned we have an enormous resource, not just with us. We don't bring all the research to our own center. We've got 300 faculty in our College of Ag. I can connect you with them. I can connect you with them. Connect you with them. We just want to have them in our. So we actually encourage that sort of feedback to say. On the other hand, for example, this SeedX thing. They contacted me, I got interested in it, and now I'm out sort of trying to scare up industry people who see you have an application. I mean, they may not want to take the time to try it just all on their own, but if it's there, they can use the machine, give it back, see if it works. If it does, they'll get their own. But if it doesn't, it's not that big of a deal. So it goes both ways. Sometimes we have very exciting research we think will apply. A lot of times they come to us. We mentioned gene editing. Everybody said, gene editing, that's easy. It's not in most plants where you don't have a really good regeneration cell culture type system. You know, Rabbidopsis, tobacco, it's easy to demonstrate the technology very quickly. But in many of these crops, in cotton, one variety that you can transform. Peppers, one variety you can transform. So if you are gene edit, you have to do the same thing almost. So when you get it, you have an enormous amount of back crossing and so on to make that a viable variety. And so a lot of work in many crops to get those haploids. We have a lot of crops who might like to make doubled haploids for breeding. Those tools and those technologies are not available in all the different crops, and certainly not in all the different genotypes of different crops. So they're willing to support fundamental research in those areas that will enable them to move those technologies along as well.
Concerning your efforts about trying to pin down the regulations that exist, the barriers that exist to expand grafting techniques, so to speak, what potential do you see? Is the government really interested in trying to deregulate these type of applications? cheap to do the gene editing, but taking that to a market costs tens of millions of dollars. It's the bottom of the
Yeah, well, the question is about the regulatory situation and how is it going to be going forward. And, I guess I can talk first about the U.S. I have to be optimistic that, in fact, they have more or less deregulated gene editing. Basically, what it says, if you have done something that you could have done by regular breeding, you're using genes within this breeding pool, you're making small mutations or deletions, kind of like chemistry or radiation can do, we're not going to regulate it. So that opened up a lot of opportunity. But my own view is that, of course, that will work great, but it's kind of limited. You can't make a BT insect-resistant crop without having the BT gene from a bacteria. But you can make herbicide resistance by modifying an existing enzyme, for example. So there's, it's plus and minus. The biggest question right now in the seed industry is if they use that, what are they going to do? Who are they going to tell? Our regulatory agency is very nervous about people doing that and just putting stuff out. They're afraid something bad will happen. The public will come to say, why did this happen? And they go, well, we didn't know about it. Very embarrassing for the government, regulatory agency. We didn't know anything about it. So the dilemma is, if you make one of those products and you market it, should you be on a list somewhere? Should you tell somebody? It won't have to be labeled, but it would if it went to Europe, for example. So the lack of harmony in the global situation makes it still very hard to commercialize. I thought that myself that in the vegetable area, that the American market would be big enough that companies could breed specifically for that and use some of those technologies. But in talking with them, many of them are so global. They grow seeds everywhere around the world, their markets everywhere. They're really still quite nervous that something will get into their system that will show up somewhere and cause a big problem. Anybody know about the orange petunia? Last year there was an orange petunia showed up somewhere. It turned out was a transgenic plant. Someone had started breeding with it. It was in petunia varieties of all the major flower companies nobody knew. It wasn't all orange. It brightened the colors of everything. Everyone had used it not knowing it was transgenic. Everybody had to recall everybody. So they're scared about the liability. So the decision right now, I think, is kind of mixed. The big, even the really big companies in the transgenic world are concerned. They're going to use gene editing as much as they can. They still don't really know what to do about the external market. They're going to say, yeah, we use that. But then the next big problem is that, like in China, you have to get regulatory approval in your country, your home country, before you ask for regulatory approval from them. Well, if this is a product that does not require regulatory approval in the U.S., there's no way you can make that application. In other words, many other countries rely on the package of data that's submitted first in the United States as a basis for going forward. If it's not regulated at all, there's no package, and then there's no way to go forward without doing all of that work yourself again. So I think I'm being cautious. I think I think that we will see those products showing up, gene-edited products, first by the big companies that already know how to do transgenic regulation. But I think we may also see products showing up from small companies that didn't tell anybody, and then you'll have people, you'll have groups that will criticize that. I'll just put it that way.
Much of the position sometimes isn't so much procedure such as the pilots in the hands of big companies. Yeah. And what about trying to engage in conversation with small companies, those who work organically, for example, and try to convince them that by just taking resistances from the same species and putting it into the varieties will make it organic friendly, for example. Why not use that strategy? Has it been tried, or people just shut their ears and don't want to hear anything about it? Or?
Yeah, well, I'll repeat the question, the comment. Could you make products, for example, that would be attractive to organic and do that? We actually tried to do that very early, 25 years ago, because our student farm on campus wanted to grow heirloom tomatoes, very old varieties that were susceptible to nematodes. They couldn't really grow them on the student farm. Another colleague there had cloned the nematode resistance gene in tomato that all tomatoes in California have. It came from a wild type. We said, well, we can put that in all of these heirloom tomatoes. It's still an heirloom, but it'll be nematode resistant. The problem was that we didn't have the money because you have to because they're transgenic, you have to be in the greenhouse. If you put it outside, you get huge borders. You know, the quarantine is very expensive. In fact, now we have a project, or we're collaborating with others who have a project funded by Pioneer to do exactly what you said. They have licensed the CRISPR technology, and they're really trying to make it open source to other groups. So they're telling us, if you can identify traits that would be attractive to organic growers, that are CRISPRable, meaning we can fix it with CRISPR, they are helping support that, and they will make the plants. Before, all we can do is talk about it. Now it's simple enough. Let's just make the plants. Let's grow it in the field, and then you say, do you want this or not? Would you like to have that or not? I mean, we moved a bacterial spot resistance gene, or we helped the group, it wasn't all our work, from pepper into tomato. One gene into tomato that would prevent the application of huge amount of copper-based fungicides or bactericides in Florida, where they grow fresh market tomatoes. But we could never get it through regulatory because it's too costly, and the industry was not willing to go with a full GMO type regulation. So that group is now trying to repeat that with gene editing and see if the industry will take it for that. So I think we'll see those cases showing up where they'll be presented with a plant, an accomplished fact. Here's the plant. Here's the resistance, or here's the trait. Do you want it or not? It's a big debate in the U.S. right now on organic. The Organic Advisory Committee considers gene editing a GMO, but the national program has not officially ruled on that yet. So it's not really clear whether it's going to be allowed in organic or not. So,
I have one question.
Yeah.
So you told us a lot about collaboration with companies, but I want to know how does the university handle innovation and ideas that are born in the university, for instance, by students, and they want to make a company out of it. Do you have like a incubator or?
Yeah, it's a very good question. How do we handle intellectual property, and can we move that on and so on? This is always the first question in these types of collaborations. If a company funds research at UC Davis, in most cases, UC Davis will own that intellectual property. So that's the first hurdle. They have to agree to that. If they want to own the intellectual property, they have to pay a much, much higher overhead cost. It's usually not worth it to do that. So generally, when they agree to do research with us, they agree that the new intellectual property will go with the university. Of course, what they bring to it, their germplasm, their ideas, or what they have, they probably already patented. So they of course keep that. But the university will own the new technology. They will have the opportunity to license it back, because we're not going to commercialize it. So that's the deal. So if they're not willing to have that agreement, when it's a consortium, that works fine, because none of them are going to own it individually anyway. For example, the genome work is just going to go out. As far as the development of a company after that, we have a number of companies coming out of the campus, but we don't really have a full-blown system to really support that. We still need to work on that. We have a group that's very strong in innovation in the business school, so they are offering programs in innovation. We have a thing called Venture Catalyst in our corporate relations and research office, which really promotes that. What we don't have is a real space on campus where you can start up companies. We're very limited in space in our region, in the Central Valley, for just space for these types of innovative companies to get underway. But we're working very hard to make that space, where good ideas that come out of a lab and somebody graduates would like to do a startup rather than a postdoc. We need that opportunity. We don't have we have some parts, but not all of it right now. But we would love it because the intellectual property owned by the university, if someone doesn't take it to commercialization, we don't get any money out of that either. We really need that to be carried forward and generate royalties, or why have it? Why have a patent? Good question.
One last question. You talk about dry chain. What about vacuum packing in bulk? I mean, it's quite cheap and it should be very readily available for everyone. What about that technology? Is it high potential to be used in a bigger scale?
It's a great comment, and in fact it is. It's something we've known. It has two advantages. One, it keeps it dry because you have to have impermeable packaging. Also, it lowers the oxygen, which helps longevity of seeds as well. And in fact, there is a company, a colleague of mine that works in this drying project is working with them. It's a Dutch company that has developed bags and so on with a simple connector and a little vacuum pump. So it's not very hard to pump vacuum. Apparently the trick was getting an easy connector that would work and would be in. And so what they have is a little instrument machine that you can plug in, say up to pretty big size, or even a warehouse, I guess, but you think of a 4x4, a meter by meter, 1 meter bin. Put it in a bag, you put it on, and you can either evacuate it or you can replace the air with nitrogen. Take half of it out, put nitrogen back in, or CO2. I mean, you can change the atmosphere. I think it's a great idea. I first visited a major seed company in Thailand, hot and humid all the time. They were trying to keep a warehouse the same condition we have in here, but drier. Cool and dehumidified in Thailand. Very difficult. And they were spending an enormous amount of money on the dehumidification. They were trying to just have the seeds inside the warehouse be kept. It makes a lot more sense to put all those seeds in plastic bags and just worry about the temperature and not try to dehumidify. The problem, of course, when you cool anything in a humid climate, it gets wetter. I mean, you have the problem with the moisture. But if you've got it really dry and then packaged, so I think the first thing is I still think the drying is the most critical. You have to get it dry. And then two, if you can put it inside a bag and put the vacuum on it, those seeds are not going to get into the next pile. They're clean, they're secure, and you can see immediately that it's not leaking. I mean, it's vacuum. So I think that's a great technology. For the longer term, we were also talking with groups like the big grain shippers who are shipping huge amounts of commodities who are taking no effort whatsoever. We grow almonds in California. We dry them very carefully to prevent aflatoxin. They go on a big freighter. They want to go to Europe. Where do they go? Immediately to the Panama Canal. As they go through there, it's quite hot and humid. Moisture content increases. A few days later, they're in the North Sea, and it's cold. So you have moisture, you have the humidity go up again, and it shows up, oh, you got fungus, you got aflatoxin here. It left us, it was fine. And surprisingly, I find that the shipping industry does nothing about this. It's really not on their radar that they could seal those bins. If it's dry when it went in and it was completely sealed, it would be dry when it came out. You can't allow the moisture to it. So that's why we're focusing really on the packaging as part of the solution. Yeah.
So, have we done some screening on vegetables to assess some QDLs related to the regeneration capacity of some lines on future culture, for vegetables, and try to...
Yeah, the question is, have we done genetic studies that identify genes for regeneration? So, yeah, we've done the most work on pepper. So we've been funded by one of the seed companies to work on pepper, which has, again, one variety that is regenerable easily and transformable, but it's not easy in others. That's only a cayenne type. So we have done that. It's very tedious, but we've done QTL analysis on regeneration capacity. So we've created the populations, tested them in regeneration, very slow process because it takes a long time. But we did find some genetic loci that seem to be associated with that trait and so on. And we learned a lot about the techniques as well. Most regeneration techniques are just sort of what somebody came up with. They're art. It just happened and it worked. We don't have a real rationale for why it worked. So we'd like to, that's a project where we work to a certain point and the company decided that's good, we'll take it internal, and I don't know what they're doing with it now. But, you know, it is clear that there are traits for regeneration, I mean, genes for regeneration ability, and that's probably what we need to do. If we can find those in the lines that are highly regenerable and cross those into what we want, that would make our life easier. But yeah, I think it is possible. We've done certain parts of that. Thank you for staying. I see it's quite late for our discussion.
Thank you once more. I think that thought was highly interesting, and also all that discussion. I think we can keep on talking more and more, but then, you know, Kent will be with us all this week, so if you want to discuss something further, just send me a message and then we can continue. Something just a final remark is that one of the sort of conclusions that we got from our previous academic advisor that was a couple of weeks ago with us,
So they can hear you.
it was this communication part, that is like one of the strategic objectives that an organization should have, and to make it also simple and very straight to the people, so they really get into the importance of the job that people is conducting. Well, thank you once more, and let's keep it.
Thank you all for having me. Appreciate all your questions. Very good questions. And I will say, this is just an introduction. I hope some of you do have specific topics you'd like to talk about. I can talk about any of these in more detail.